Welcome to this podcast edition called Chromosomes. I hope that you'll get a lot out of watching. The main goals for this podcast are to, number one, describe the types of chromosomes that carry genetic information. Secondly, compare and contrast genes that are linked with those that are unlinked. And thirdly, you'll be shown how changes to the physical parts of a chromosome and extra or missing chromosome pieces affect the organism. Here's the format for Cornell notes taking that I'll ask that my students follow. The title of the chromosome lecture is at the top there. Make sure that you're writing your questions in the Q column along with main ideas and topics, and then don't forget to do the summary, please, at the end. Some questions that will drive this podcast and others related to it are listed. Number one, how are traits passed from one generation to another? Number two, why do members of the same family have different traits? For example, you and your siblings, if you have them, why do you look different from them? And then thirdly, why do some traits skip one generation and show up in the next generation? So this has to do with the presence of recessives in your family. You may have a grandparent with blue eyes and your parents have brown eyes, and if you have blue eyes, then that's an example of the blue-eyed recessive trait skipping your parents. Some words that you want to be familiar with before we proceed are listed here. So a chromosome is basically a packet of genetic material of DNA that is passed from parent to offspring through their sex cells. In humans, the diploid number is 46, so we have 23 pairs of chromosomes, one pair from one mom and one pair from the father. And then the homologous chromosomes, what that means is that you have a mom set and a dad set. So that for every chromosome, for humans, numbers 1 through 23, there is a duplicate set. So together they're called homologous chromosomes, or sometimes they're called homologs. A gene is a short section of DNA that codes for a given protein, which turns into a trait like eye color and hair color. An allele is just a different form of a gene, so it's like a gene letter, dominant versus recessive. Sex-linked genes are those genes that are located on either the X chromosome or the Y chromosome, which is the 23rd pair in humans. And certain genes live on those chromosomes, like pattern baldness, hemophilia, and for men on the Y chromosome, the gene that makes boys boys and not girls as they're developing as fetuses. Crossing over is a process that happens during meiosis, where bits of one chromosome are exchanged with bits of another chromosome, thereby making sex cells with unique chromosomes. Independent assortment describes the fact that any two traits that are not linked together on the same chromosome will separate. So, for example, in pea plants, if you're looking at plant height, tall versus short, and the color of the pea seed, yellow versus green, then those two traits will transmit from parent to offspring independently of each other. A mutation is a change in the DNA sequence that results in a different protein and a different trait. A lot of times recessive genes or traits are produced this way. And then aneuploidy is basically a situation describing a cell whose chromosome number is different than that of normal. So to start off looking at chromosomes, on the picture on the left you'll see kind of a clustering of chromosomes. This is what it would look like if you were to break open the nucleus of a cell, and you would see these chromosomes of different lengths. You'll notice that they're all X-shaped. You have one chromosome that has been duplicated, and so the other side of the X chromosome is actually just a copy. And you'll notice in the cartoon picture on the right that genes are actually smaller than chromosomes, and genes live on the chromosomes. This is the basic breakdown of how genetic material is arranged. So on the largest scale, we have a cell, and if we break down the cell, we look at different organelles, including the nucleus. If we sort of break open the nucleus, we have chromosomes. If we examine chromosomes more closely, we'll see that genes are located along its length, and then a gene at its very basic level are basically DNA letters, what we call nucleotides, A, T, C, and G, shortened for adenine, thymine, cytosine, and guanine. So when we look at the chromosome, there's two basic kinds of chromosomes, those that belong to bacteria, or they're sometimes called prokaryotes, and every other kind of organism is called eukaryotic. And bacteria have a circular band of chromosomes, like a loop, and in our case we have linear chromosomes that form into the X-shaped chromosomes that you'd seen in the previous slides. So if we look at this graphic and we go from right to left, the chromosome is shown in its classic X-shaped form, and if we were to unravel it, much like taking a sweater and unraveling it thread by thread, you'll notice that it goes from a large scale to a small scale. The smallest scale, you'll see the DNA, which is the blue loops, are actually wrapped around these purple structures, and those are just proteins. And so the purple proteins are meant to provide kind of a scaffold for the DNA to wrap around, and it also provides protection so that the DNA is not damaged. So there's three different aspects of the chromosome theory of inheritance. Number one is that chromosomes and genes are present in pairs as diploid cells. The other part of the theory of inheritance says that homologous chromosomes, again those chromosomes that are obtained from parents, they separate during meiosis. So at the end of this process, we'll actually have daughter cells, which we call sex cells, that are haploid. So they contain half the number of chromosomes as the parents do. So we look at chromosomes, and there's sex chromosomes. That's the unique pair of chromosomes that determines males versus females. And so XX is the sex chromosome pair for females, guys are XY. And then if it's not a sex chromosome, then they would be autosomes. And autosomes would be the other 44 chromosomes, or chromosome pairs 1 through 22. Sex-linked genes exhibit unique patterns of inheritance. So for those genes that live on the sex chromosome, they have a different pattern of inheritance, because whether or not you're a male or a female, that determines very greatly your chances of getting the trait. So if we look at the sex chromosomes, this is actually the Y chromosome pictured, and it's much shorter than the X chromosome, and there are just a handful of genes on there that geneticists find notable. One of them is the SRY gene. That gene, when working in a male fetus, will actually determine that it's a male, and so the male sex parts will be determined and produced from that. So girls lacking this will actually become girls. So here the system for determining gender is shown. For humans, the gender is determined by two X chromosomes determining a female's gender, and then XY is the male's sex chromosome pairing. So where it says 44 plus XX for the female, that's 44 plus 2 sex chromosomes, which leads to the 46 chromosome number. And then you can see the same sort of thing for the males. In grasshoppers, you'll notice that they have a different number of chromosomes. Every species has a unique chromosome number. In this case, their number is 22 plus 2, which is 24. Here, the female is determined by two X chromosomes paired together, whereas the male is determined by just a single X chromosome. So there's no second chromosome there, so there's no Y in that concept. For roosters and hens and chickens, you'll notice that they have a total of 78 chromosomes, but their female gender is determined by having two sex chromosomes that are different, whereas the males are determined by having two sex chromosomes that are the same. So it's really kind of the opposite than we see in mammals. And so instead of female mammals being XX, in this case, it would be XY. That would be the equivalent. For bees, you'll notice that there is a different system. Here, the diploid cells would determine the female, and the haploid cells would determine the male. So no concept of X versus Y. It's just the number of total chromosomes. The third part of chromosomal theory of inheritance says that fertilization restores chromosome number. So you have two cells that are haploid, so that would be the sex cells, that would be sperm and egg, you'll notice at the top of the picture. And when those fuse, they turn into one cell that is diploid. So it's very important for you to understand what this graphic shows. because this is really the cycle of life. So fertilization brings two haploid cells together to make a diploid zygote. Again, a zygote is just a fertilized egg cell. In this case, it's diploid because it contains two copies of the 23 pairs. And then when that diploid zygote matures and grows through mitosis or through cell division as reproductively mature individuals, females will produce egg cells in the ovary, males will produce sperm cells in the testes, and then those will be the raw material for the next cycle of life. So we've talked a little bit about chromosomes. There are sex chromosomes, there are autosomes. Male and female in humans are determined by different combinations of the X and Y. And we understand that there are some traits that are linked to either the X chromosome or the Y chromosome, and those are called sex-linked traits. In the early 1900s, there was a gentleman by the name of Thomas Hunt Morgan, and Dr. Morgan studied fruit flies. And in this picture, you'll see that there are two kinds of flies. One on the left has red eyes, the one on the right has white eyes. And so in the way that he studied it, the language that he used was dominant or normal is wild type, and the mutant is the recessive. And so in this case, this white-eyed fly on the right is actually a male. What they had found in their experiments was that if they took a female who was red-eyed and a male who was white-eyed, all of their offspring would look like the mother and have red eyes. If they took those F1 red-eyed flies and they mated them, the third generation, the F2 generation, would have kind of a curious result. All of the females would look like the parent female in generation 1, but half of the males would look like the wild type red-eyed and half of them would be white. So the question is, why is that happening? And at the time they didn't understand what sex-linked traits were. So this is a classic experiment that led to the current understanding of sex-linked traits. So the conclusion basically is described as such. And so you'll notice that the female on the left has 2 X chromosomes, whereas the male only has 1. You'll notice that there are W's that are attached to the X chromosome. The W plus is just the dominant wild type gene, and the W without the plus is the mutant recessive. And so in this case, the white-eyed male is white because the only X chromosome that he possesses is the recessive variety, and so he has no choice but to express the white-eyed trait. Whereas the female could be fully wild type, or she could even be heterozygous and still have the red eyes. So when the parent generation is paired together, they'll show that their offspring are as follows. So all of the females are hybrids or heterozygous, one wild type and one recessive allele, whereas the male has, in this case, the wild type allele from the mom. And when F1 generation is crossed and they produce F2 generations, you'll notice that the one white-eyed male has the recessive mutant W allele. So that's how the reasoning of sex-linked traits was discovered. So summing it up, Gregor Mendel proved that genes exist on specific chromosomes, and when chromosomes are passed from parent to offspring, the genes also are passed. Thomas Hunt Morgan proved that specific genes, a small handful of genes, exist only on the sex chromosomes. In this case, the color of the eye for fruit flies is located on the X chromosome. In humans, the X chromosome has most of the genes, many more that are unrelated to sex, whereas the Y chromosome has just a few genes that are found on it. Some jokingly say that the Y chromosome has genes that make men like football and be able to use the remote control well. You can make your own determination. If we look at X-linked genes, there's basically 3 of them that are classically studied. One is hemophilia, which is the inability to clot blood. The second is pattern baldness. And then the third would be color blindness of various varieties. And so in this graphic, we can see that the father is XY and the mother is XX. He is unaffected, so his allele on the X chromosome is the dominant version. The mother's the carrier, so she has one dominant and one recessive allele, noted by the white and the blue squares. So she does not have the the trait in question, but she has the ability to pass it on to her children. And so when you look at the children, 2 sons, 2 daughters, the affected son receives his recessive allele from his mother, and that is why X-linked traits are always passed maternally from mother to son. Here's 3 scenarios that kind of show how the various ways of transmission occur for sex-linked traits. So in this case, the white square represents normal, the orange square represents the affected individual, and then the tan square represents those individuals that are carriers. So you'll notice that when you pair a normal female and an affected male, half of the children will be carriers. None of them will have the disease or the trait in question. The females will be the carriers and the males will be normal. In the second scenario, we have a carrier mother paired with a normal father, and half of the girls will be carriers, half the boys will be normal, and one of the boys will actually have the recessive trait, whether it be hemophilia or color blindness. In the third and most serious condition, you have a carrier mother paired with an affected father, and so in this case, half of the children will have the disease, 1 girl, 1 boy. The other girl will be a carrier, and then the other boy will be normal. So these are some of the disorders that are caused by recessive alleles on the X chromosome. So now we look at genes. So genes exist on chromosomes. Genes can either be unlinked or they can be passed on to children independently of each other. They have no effect, or they can be linked. In addition to that, genes follow the rule of independent assortment. So genes for any 2 traits will pass on independent of each other as long as they're unlinked. And that there's this crossing over business that happens during meiosis where bits of chromosomes are exchanged from one sister chromatid of one homologous pair to the other. The result of this would be new gene combinations. So if you have a swapping of genes, then the resulting sex cells will be different from each other, and then when those are fertilized, they will produce offspring that are different when compared to each other. And then the long story short would be that new phenotypes or traits are possible in this process. Sometimes, if things go badly, genetic disorders can be created, and then possibly, if they are severe enough, death. Let's take a look at how independent assortment works.
During metaphase 1 of meiosis, each joined pair of homologs lines up on the metaphase plate. The orientation of each pair on the spindle axis is random. Either the maternal or paternal homolog may orient toward a given pole. The number of possible chromosome orientations equals 2 raised to the power of the number of chromosome pairs. In this hypothetical cell with 3 chromosome pairs, 8 2 to the 3rd possible orientations exist. Each orientation produces gametes with different combinations of parental chromosomes.
So if we look at this last graphic, you'll notice that I've labeled the various chromosomes with either a capital or a lowercase. And you'll notice that the random nature of which of the versions, the dominant versus the recessive, line up on the left side or the right side of the cell will determine what the resulting sex cells will contain. So in this case, we have 8 cells that are the various combinations of where the chromosomes line up. In our case, we have a total of 8 million different combinations that can be produced based on just which side of the midline of the cell a particular chromosome will match up. So there's a lot of variety that comes from independent assortment. So crossing over helps to increase genetic diversity, and then independent assortment does as well. And then lastly, the random nature of which specific sex cell, egg versus sperm, fertilize each other, that also is another layer of diversity. So there's lots of different ways to create many, many different genetically unique individuals. Linked genes tend to be inherited together because they're located near each other on the same chromosome. So of the total 25,000 or so genes that are located on in our cells, they're spread over the total 23 pairs of chromosomes. And if those two any two genes are linked, they're going to be found on the same chromosome, so they'll actually be inherited together. Lastly, we'll look at the number of chromosomes and their structure, so things that we can do to their links or the arrangement of the genes that are on there. So there are structural problems, and those structural problems include segments of the chromosome being duplicated or doubled, parts of them being deleted or removed entirely, of switching of chromosome sections between chromosomes, and then inversion, meaning that a section of the chromosome is actually flipped 180 degrees. In terms of number problems, there is a concept of aneuploidy where through meiosis there's not an even splitting or distribution of chromosomes from the parent cell to the resulting daughter cells, the sex cells. And if that happens and those particular the sex cells are fertilized, then you can have various genetic disorders that are inborn. For example, Down syndrome versus Turner syndrome. Here's what we call a karyotype. It's a picture of the chromosomes that are found within the nucleus of any given cell. There's a commonly done genetic test that's done before a baby is born to determine if the individual has too few or too many chromosomes. So this karyotype can help determine if a fetus is affected with Down syndrome or some other genetic disorder even before it's born. So you'll notice that each of the 22 numbered pairs are the same length, and then you get to the X chromosome, and the Y chromosome is the 23rd pair, and the Y chromosome is much shorter. There's not as much DNA there to hold the genes in question, but they are considered to be the sex chromosomes. So you can look at a chromosome karyotype like this and see if there are too few or too many chromosomes. Now, if we were to vary or change the nature of a chromosome, each of these little blocks, A through H, for example, in scenario A, represent just a segment of the chromosome. And within those segments, there may be one or more genes for a various different traits. So in A scenario, we have a deletion. So on the left-hand side, the D segment is there, and it's been deleted in the resulting segment on the right. In the B scenario, we have the BC box, which represents a section of the chromosome being duplicated. So in the right-hand scenario, it's actually doubled, and that can be a problem. You can see that in scenario C, we have a kind of a flip where you have BCD. In that sequence has actually been reversed. Now it's 180 degrees different, and now it's DCB. In the last segment, we have two chromosomes on the left. We have chromosome A through H, and you have chromosome M through R, and you have an uneven swapping of two sections AB swapped with three sections M, N, and O. And so when you see the result, you'll notice that the one chromosome is actually shortened. The bottom chromosome actually has a total of 5 segments, but it started out with 6 segments. And so these are different ways that the chromosome can actually be changed, and many of these can result in some very bad effects. In terms of the number of chromosomes that are passed from parent to offspring, this would be an example of Down syndrome. So the 21st chromosome that you can see circled is actually called trisomy. It's 3 chromosomes. And it's that extra chromosome that actually leads to various effects of people suffering with Down syndrome. So how does that chromosomal math become messed up? So non-disjunction is the mechanism by which the chromosomes are not separating. So you'll see in this graphic the parent chromosome at the top is actually split into 2 cells, and the chromosomes should split evenly. But in the first stage, the chromosomes did not separate evenly to either cell. So you had extra chromosomes resulting in the cell on your left, and you had too few chromosomes resulting in the cell on your right. And if you follow that through to production of the gametes or sex cells, you'll notice that 2 of them have an extra chromosome and 2 of them have a missing chromosome. So if any of those chromosomes, any of those sex cells were fertilized, you would have potential for a chromosomal problem in the offspring. In the second scenario, non-disjunction can happen in stage 2. You'll see here where the chromosomes are not evenly split in the first cell, noted by the non-disjunction box. And then you'll notice that the sex cells that are being produced, the gametes at the very end, half of them, the third and fourth cells, will actually have the normal correct haploid number. And it's the first two that are producing the aneuploid number, n+1, n-1, but not n. And if those first two sex cells were fertilized, then you would have the potential for a chromosomal problem. So that ends today's podcast. I'd like for my students to answer these 3 exit task problems that are shown here and 2 following. Well, thanks for joining. Today you should have seen how chromosomes are grouped together as either autosomes, of which there are 22 pairs in mammals, or sex chromosomes, which are a single pair. Sex chromosomes carry genes called sex-linked genes. There are only a handful of them, however. Most famously, the X-linked traits include color blindness, hemophilia, and pattern baldness. Secondly, since genes live on chromosomes, any 2 that live on the same chromosome are considered to be linked, but any 2 that do not live on the same chromosome are unlinked. And then lastly, chromosomes, which are made of DNA, can be changed as a result of gaining, losing, or switching segments. And furthermore, when an entire chromosome is missing or duplicated, often serious genetic disorders like Down syndrome can occur. Thanks so much for joining, and I look forward to seeing you next time.